right, here we go. We're going to continue on with our cell communication and cell cycle notes. I will tell you that in my rush to grade your tests last night, I didn't get to finish this presentation. So I'm going to have to, I got cell communication done, but then the cell cycle, like mitosis and cancer, I'm going to go to our original PowerPoints and just zip through them with you really quickly. I'll skip the slides that I think aren't important or whatever. Okay, so just reminding you, we're talking about cell communication first and about signaling pathways. And there are 3 words that I want you to try to remember. And if you can just have those words, you've got a good start, which is just the letters RTR, reception, transduction, response. And if you haven't written those down, I recommend writing them down. That's basically what's happening when we when we're talking about cell communication.
The trucks and stuff?
Say this again. Nope, that's DNA. The trucks are in DNA. So we'll talk about that a little bit later. So signaling pathways are widely conserved among all domains. That basically means that all organisms share these pathways. So that's an indicator, of course, of common ancestry, and that these pathways evolved very early in Earth's history and in life on this planet. Okay, so the things you need to know. So there's the same image. Do you see that that's the same image? Mm-hmm. Like your eyes are going to have to get good at looking at totally different images and being able to be like, okay, there's the RTR, the reception, the transduction, and the response. So we'll start with reception. Remember that the signals, they look like a guitar pick here. The signals are called ligands. And the ligands can be any of many different types of molecules. Some of the ligands are hydrophobic. And if they're hydrophobic, we've got somebody with the hiccups in here, so you'll hear a little percussion every once in a while.
Sorry.
It's not a problem at all. So the hydrophobic ligands, they can actually pass through the cell membrane. So between the phospholipid bilayer, those ligands can pass right through. If they're hydrophobic. If they're hydrophilic, they can't pass through, so they need that membrane protein. So in this case, what we're looking at is one that's hydrophilic. Because it didn't pass through the cell, it had to use a protein to send its message through the cell. Okay, we'll see one that's hydrophobic in just a moment. All right, and then it says changes in the ligands, so the guitar picks, or changes in the receptors, the purple proteins, alter the responses. And so what would cause changes? Well, the easiest one is mutations in DNA. Mutations in DNA are going to change the shape of the protein, and so then that cellular response, that cellular pathway, the signal pathway, is going to get screwed up. But coming in contact with chemicals in the environment could change the ligands. You might come across a ligand that tricks your body into thinking you produced that ligand when you really didn't. Those are examples of things that could interfere with a signal pathway. Here's an example of local cell signals. So sometimes the cells are communicating directly with one another. This is an example where we have like a white blood cell that is traveling around the body, and it's touching every single cell, making sure that that cell doesn't have any invaders. And if it does, then it attacks. So a white blood cell touching the surface is an example of a local cell signal. So that's enough like talking about the signals themselves. So now let's talk about the transduction. So once that, how do we get the message into the cell, and then what happens? So the last part is response. We're going to talk about within the cell itself. So you might remember this. It's definitely on our learning targets. I would say review it like right now, review it with me, and then never think about it again. Like it's not worth your time to spend hours trying to memorize it. Be familiar with it, look at it in the diagram, be able to identify what's going on in the diagram, but don't do any memorizing of it. So they're called G protein coupled receptors, or GPCRs. I always have trouble with the order of those letters. Actually, PCR, I should think of that because I know what PCR is. We learned about PCR, so GPCR. So it says how vision and smell are transmitted through neurons would just be an example. They use the GPCRs. So the ligand in this case, I'm giving you an example, would be a neurotransmitter, and it activates the GPCR, changing its shape. So take a look at the diagram, look at the ligand. It attaches to the receptor, and then the molecule down below. So it says the interior of the GPCR now activates a G protein. So notice that G protein has been activated, the little teal molecule that's inside. It's been activated. And importantly, it uses GTP, not ATP. That's why it's called a G protein. It's because it's using a different kind of molecule. And then it says the G protein then activates an enzyme. Look at it. That triggers cyclic AMP production, which is a second messenger. So then it mentions, I think this one mentions, maybe it doesn't.
Did we do that? Oh, no.
I was wondering if it maybe mentioned a phosphorylation cascade, if this diagram mentioned it, but it didn't. We'll get to that in just a minute. So other examples of transduction, this is a ligand-gated ion channel. So there's the guitar pick, there's the ligand, and notice that there's a flap on the protein that is closed right now, and it's not allowing the ions into the cell. But once that ligand is present, it alters the shape of the protein. It's not that it's a gate that opens and closes, it's just that the protein changes shape and now it's in an open conformation, and now those ions can come through. And at any time that that ligand leaves, then the shape closes up again. And so the ions can't get through. Would that be in this diagram? Is that a hydrophilic or hydrophobic ligand? That is a hydrophilic ligand because it's requiring a receptor. It can't get through the membrane on its own. So good. Moving on. Okay, so the second messengers, this is mentioned in your learning targets. Just what are second messengers? They're found on the inside of the cell, and they trigger additional response pathways. Sometimes they trigger the phosphorylation cascades, which we'll see in just a second. But cyclic AMP is one of the most famous ones. What do you need to know about cyclic AMP? Pretty much nothing. Just don't let it freak you out when you see it. So cyclic AMP is just continuing transduction. We're moving the message in the cell. We started it outside the cell. Now transduction means to move the message, and we're going to get to the response in just a second. This is kind of one step in transduction. This is the phosphorylation cascade, where we have a whole bunch of proteins that use up ATP. So look at the diagram. Do you see where, I'm going to point to it on the screen here, the ATP gets used up? So here the ATP gets used up, and we add a phosphate, and that changes its shape. And then again, we add a phosphate and it changes its shape. Every time you add a phosphate, that changes the shape of the molecule. And what's happening in the phosphorylation cascades is amplification, where we're going from one tiny little message that arrived in the ligand. It's one tiny message. And by the time you get done, you have a million messages that have all been sent. So it allows cells to respond incredibly fast. One message, whoosh, and we've suddenly inundated the cell with this particular message. So from a single ligand, a phosphorylation cascade can amplify the response tremendously. I wrote tremendously. Okay, and then what are examples of cell responses? What is it that we're even trying to do? We're sending a signal, it's moving through the cell. What's the purpose? So cells need to respond to light, to temperature, to hormones, to senses, to stress. How do they respond? You don't need to memorize these, but just be, well, it might be nice to have a couple of them in your back pocket to even say. For example, it might, whatever. So turning a gene on or off would be an example of a cell response. So making a protein or not making a protein anymore, that would be, if you turn a gene on, you'd make a protein. If you turn a gene off, you would stop the production of that protein. Activating or inhibiting an enzyme would be an example of a response. Telling the cell to divide or not, that's a response. So mitosis, remember sometimes mitosis is bad in the case of cancer, so we don't want too much mitosis. Going through apoptosis, maybe there's something bad going on with this cell and it needs to suicide. So apoptosis causes the cell to die. And there's many more possible responses. All right, epinephrine was just one example. It's polar. We can see that it's polar because, or hydrophilic is another way to say it, because it needs a membrane receptor. And then it is mediated by GPCRs. So you see that it changes shape, and we have the G proteins using GTP. We see cyclic AMP being used, and we get all kinds of responses from it. In this case, the inhibition of glycogen synthesis and the acceleration of glycogen breakdown. Do you need to know that? Nope, don't need to know this. This is just an example that they could ask you to interpret the diagram or whatever. You would never need to pull this out of your back pocket. All right, and then signal transduction and disease. A mutation can change the shape of the proteins, and so that could lead to any kind of a disease or cancer, for example. And then just a quick reminder of negative and positive feedback. We're almost to the end of this section. Negative feedback, remember, is decreasing the stimulus. So the example here is when the body temperature is going the wrong direction, either too high or too low. We want to return it back to the normal. So it stops a condition that is detrimental and it returns the body temperature back to normal. That's negative feedback, stopping. Positive, oops, I thought I had positive feedback on here. Did I miss it? Dang, I didn't have my positive feedback. So positive feedback would be an example where we want to continue and make it happen more and more. And the classic example of positive feedback is pregnancy and birth. And when the baby starts to push on the cervix, it releases a chemical called, not the baby, the brain releases a chemical called oxytocin, which triggers labor. It triggers the pressure of pushing, which causes the baby's head to push more on the cervix, which triggers more oxytocin, which triggers more labor. And so it keeps going and going and going until the baby is out and no longer pressing on the cervix, and then labor stops. But the positive feedback, it continues as long as that baby is continuing to push on it. Okay, so I'm going to stop here because I didn't get any further with this particular review set of notes. So we're going to switch over to the notes that are called mitosis and just zip through those quickly. These don't take as long. Just a moment. There we go. We got both of them going. Okay, so just things why cells need to divide. Cells need to replace damaged cells. They need to grow. Asexual reproduction, so just replacing your skin cells or whatever, that's an example of mitosis, when mitosis is going through that. Okay, and then the cell cycle, just reminding you about the different stages of the cell cycle. Oops, I went too far. So in this diagram, the yellow pie piece in this diagram, that's mitosis and cytokinesis. That's when the cell is dividing. The whole rest of the cell's life is spent doing other things. I sometimes draw that analogy that a woman is pregnant for a tiny part of her life, and the rest of the time she's doing other things. So in this case, G1 is when the cell is growing. Remember that G0, the G0, is when a cell actually stops dividing. So that could be normal, like nerve cells don't divide, and so they automatically go into G0. It could be a sign that the cell has gotten old, and that's called senescence. And so it's not like senile. Yep, it's not supposed to divide anymore. But assuming that it is going to be a cell that will divide, it goes through G1, cell growth. S, super important, and I do want to make sure you remember this one. S is when the DNA divides. So when you replicate your DNA, when you get 2 copies of the DNA. So that's the place where mutations are going to take place. That's the place where mutations are going to take place, is during the S phase. So as we're copying the DNA, we make some mistakes, and that's going to get passed on to the new cell then. And then G2, it prepares for mitosis, and then the cell goes through the different cell stages. So we said this one already. Chromosomes, just reminding you about chromosomes. We have a whole review that we'll do on DNA. But just that's a chromosome, and it's got 2 sides. So the left side is identical to the right side, and those are called sister chromatids. This chromosome has already been copied. This chromosome has already been copied during the S phase. That's why it has 2 sides. That's why it's called bivalent. And it's going to get ripped apart, and the left side is going to go into one cell, and the right side is going to go into the other cell, so that those 2 cells have identical DNA. And then those cells cannot divide until the DNA goes through replication again, until it copies again. All right, I think I'm going to skip ahead on this. Oh, and just a quick reminder, the letter N, so 2N indicates that you have 2 copies of every cell. So you have, and copies of every chromosome. So you are a 2N organism because you got half of your DNA from mom and half of your DNA from dad. Does anybody remember what 1N, what cells in our bodies, the only cells in our bodies that are 1N? Anybody remember? What cells in our bodies have half the DNA? You can giggle a little bit. KJ, that would be reproductive. Nice. So sperm and egg are 1N cells. Good. Okay. Prokaryotes, they just have a circular chromosome. We have what's called linear chromosomes. So the one that looked like an X, we call that a linear chromosome. These guys have circular chromosomes. And their mitosis is called binary fission. It's a little bit different because it's a circular chromosome, but it's still the same idea. The DNA gets copied. All right. And then we did haploid and diploid. Haploid is half the DNA. Those are 1N. Diploid cells are 2N. They have one from each parent in that case. It is possible for cells to be polyploid. Maybe you remember those terms. So, all right. Yes, correct. Strawberries are polyploid. Okay, and then I don't think need to know all the details of these cell stages. Can anybody say them to me? What are the stages of mitosis? You say kind of a dumb word.
Prophase, telophase.
You didn't quite say them in the right order. What's the word? IPMAT. IPMAT. So for any mats in the room, I'm sorry that we're saying we pee, but IPMAT. So interphase, prophase, metaphase, anaphase, and telophase. Take a look at this diagram and just see if you can kind of get the hang of what's happening or could put them in the correct order. They're in the correct order right now, but if we gave them to you scrambled. In prophase, the DNA starts to twist up and it forms the chromosomes that you can see. In metaphase, they line up in the middle. Good. In anaphase, the two sides get separated. They get pulled apart. And then in telophase, you can see in this diagram, there's a tiny cell wall. They call it a cell plate right now that's forming. That's in a plant cell, the telophase that we're looking at. And so it's splitting in two. Here's an ugly word, non-reductive eukaryotic cell reproduction. That refers to not reducing the number of chromosomes. So in mitosis, when you start with 48 chromosomes and your cell divides, you end up with 48 chromosomes. In meiosis, you end up with 23 chromosomes. So that would be reductive. Meiosis is reductive, whereas mitosis is not reductive. Okay, and then I'm not going to say a whole lot more. So prophase, and then prometaphase. It's like a preteen, no, a tween. That's what they call them. Tween. It's like, that's not a real word. So prometaphase is in between prophase and metaphase. Metaphase, they line up in the middle. Anaphase apart, and telophase, they form two cells. Cytokinesis, there's our butt cheek picture. So cytokinesis is in animal cells. That entertains me no end. Okay. Animal cells are soft, remember, so the cell can just pinch in, whereas plant cells cannot. So plant cells have to have this cell plate that forms in between them. Okay, and then I think we're pretty close to done. That's a video, and we're not going to watch that. I think we're done. Okay, so we're done with that one. We've just got one more set of notes to go through, everybody. We're almost there. Okay, let's see. Cell cycle control. There we go. And honestly, this one is one that we don't need to know as much about as what I put in the notes. I put more in these notes because I think that students are interested in cancer and they want to understand a little bit about cancer. But in terms of what AP really wants you to know, let's see if I can show you. So there, you need to understand that there are checkpoints. I'm looking at so the people online can't see this right now. I'm looking at the learning targets. So what you would need to know about cancer is you need to understand that there are checkpoints. We'll get to that. And sometimes those checkpoints get mutated, and so then they don't work. Some of us are born with our checkpoints mutated. Let's see. And then cyclins, I'll just show that to you quickly. And then anytime that the cell cycle is out of control, that's cancer. So when we have like 6 to 8, it takes like 6 to 8 mutations before you actually develop cancer. So we have all the controls that we need. Should I go through it? I don't even know that I need to go. I'll just show you a couple of them just to remind you. So there's the G1. Here, give me a second. There's the G1 checkpoint, and then the G2 checkpoint, and the M checkpoint. Remember that we talked about the guardian angel gene. It's a tumor suppressor gene. And its job is to make sure that the cell is dividing correctly. And then if it identifies anything that's not dividing correctly, then it stops the cell process, and it triggers fixing enzymes that come in and fix the DNA. And then if the DNA is fixed, then the guardian angel gene allows cell division to continue. But if we can't fix it, then the cell is supposed to enter apoptosis. So there's all these things that are supposed to protect us, but something like 80% of people who have cancer, their guardian angel gene is not working. And so that's one of our enormous checks that's actually not working. I'm going to go ahead and stop because it is after 4 o'clock and people have been here a very long time. So is there any questions before we go? Anything you need? I think we're going to be done. That's it, everybody. Have a good one. Let me stop my